Okay, we now will have a brief discussion about B cells and antibodies. So B cells are also known as B lymphocytes, and they play a crucial role in the adaptive immune system. This crucial role mainly involves production of antibodies. The antibodies which are produced by these B cells, they are the ones that help to recognize and neutralize pathogens. So pathogens are things that will invade your body to cause disease. So such pathogens then will include things like bacteria, viruses, and parasites. So apart from just producing antibodies, these B cells also play a role in creating what we call the immunological memory. Immunological memory is what allows your body to respond faster to an infection that your body has previously encountered. So when you are infected today, for example, then you heal, then if you are in the same infection will again be exposed to your body sometime next year. The B cells, which will have been created and stored as immune memory cells, will help in the immunologic memory, okay? Because they will now remember that you were exposed to this infection at some point, and therefore they will be mobilized faster. So each B cell is highly specific, which means it can only recognize one type of antigen. An antigen, by now you should be knowing, is a foreign substance that triggers an immune response. So an antigen is specific. A bacteria has a specific antigen. A virus has its own specific antigen. A parasite has its own. So each of them will then trigger an immune response. So what about the development and the activation of these B cells? So B cells, they originate in the bone marrow. They still mature in the bone marrow where they develop B cell receptors. These receptors are what helps them to recognize antigens. Okay, so once they are mature, they originate in the bone marrow, they mature in the bone marrow, but once they have matured, they now migrate to the secondary lymphoid organs. So secondary lymphoid organs, I'm sure you saw that in the lymphatic system topic when we looked at that. Secondary lymphoid organs include the lymph nodes, the spleen. So in these secondary lymphoid organs, the mature B cells migrate there and await activation. So they develop, they originate in the bone marrow, they mature in the bone marrow, but then they migrate to the secondary lymphoid organs where they now await to be activated. So how does the activation of these B cells occur? So before the B cells can produce antibodies, they have to be activated. And this activation occurs in two main ways. One is an activation that happens independent of the T cells, so without the help of the T cells. This type of activation is called T-independent activation. So there are some antigens like bacterial polysaccharides which can directly activate B cells without the help of T cells. So if a B cell is activated without the help of T cells, then that is a T-independent activation. So such an activation results in a faster immune response, but that response is weaker. And it primarily leads to the production of the immunoglobulin M antibodies. The second type of activation of the B cells is the T-dependent activation. So this requires help from the helper T cells or the CD4 T cells. So we know that the antigen-presenting cells, for example, the dendritic cells, they process the antigen, then present it to the helper T cells. An activated helper T cell will then release cytokines, and those cytokines are what will stimulate the B cells. Upon stimulation of those B cells, that is now what will make them to proliferate and differentiate or change, either to become plasma cells or memory B cells. The plasma cells are the ones which will now lead to production of a larger amount of antibodies. But the memory B cells, those are the ones that will remain in the body for years to help in the immunologic memory in case there is a future infection by the same pathogen. So there is the T-independent, which does not require T cells. Then there is the T-dependent activation, which requires an intervention from the helper T cells. Once the helper T cells are activated, they release cytokines. The cytokines are the ones that now stimulate the B cells. Stimulated B cells are the ones that will now change into plasma cells and memory B cells. Plasma cells will lead to a production and a secretion of larger amounts of antibodies. The antibodies are now what will go and neutralize the pathogen or the antigen. The memory B cells will be required or will be important for immunologic memory. Okay. So let's look at the antibody structure. So antibodies are also called immunoglobulins, abbreviated as a capital I and a small g. These antibodies are Y-shaped and they are composed of 2 heavy chains and 2 light chains. So the heavy chains are longer, but the light chains are shorter. We'll see a structure of the antibody in the next page. Both of these chains, that's the heavy chain and the light chain, both of them have a constant region and a variable region. The variable region is found at the tip of the Y structure. So this variable region is what is responsible for binding antigens. So it's important that the point where the antigens bind is variable because the antigens will be different. So that position will keep changing depending on which antigen it has to bind. The constant region determines the antibody class or the antibody type. Okay, so is it immunoglobulin G? Is it immunoglobulin A? Is it immunoglobulin M? That is determined by the constant region of the antibody. Then there is a hinge region which just joins the 2 arms together so that it can increase flexibility. On page 3, you can see now a structure of the antibody. So you have, it is Y-shaped. You have the constant region, then you have the variable region, which is at the tip. Okay. You have the light chain, then you have the heavy chain. The antigen binding site is at the tip of that Y structure. Okay. Then you have the hinge regions, which are the disulfide bridges. Okay. So that is the structure of the antibody or an immunoglobulin. Okay. So it is Y-shaped. There is a constant region. There is also a heavy chain. Then there is a variable region. The variable region is at the tip of the Y shape. Then at the end of that variable region, that is where the antigen binds. So it's important that where the antigen binds keeps varying, because the antigens will also be different. The antigens that come will be different. So that increases the ability of the antibody to keep binding different antigens as they come. Okay. Okay. So what about the types of the antibodies, and what does each of these different types, what role does each of them play? So we have one that we call the immunoglobulin G. Is the most abundant in blood. Immunoglobulin G helps to provide long-term immunity, and it crosses the placenta to protect newborns. So that is then, if it is crossing the placenta, then that is passive immunity. Okay. Then you have immunoglobulin A, which is found in the mucosal areas. So because it's found in the mucosal areas, you now find it largely in saliva, in tears, in breast milk. And its main role is to protect against pathogens that could attack the body across these mucosal surfaces. Immunoglobulin M, what is abbreviated as IgM, is the first antibody that is produced during an infection. Okay, so it's the first to reach a site of infection before other antibodies can be mobilized to come. Immunoglobulin E is what is responsible largely for allergies. It's responsible for allergic reactions, and it provides protection against parasitic infections. Immunoglobulin D, it functions mainly in the activation of B cells. So it helps largely in the activation of B cells. Okay. So the mechanisms through which these antibodies function. So the antibodies function to eliminate pathogens. So how do they eliminate these pathogens? By now you should know this, but they do it through several ways. One is neutralization. By neutralizing, they block the harmful substance from entering cells. Okay. Opsonization, which means just coating a pathogen so that it can be identified for destruction. So they put a mark on the pathogen so that the immune system can identify that pathogen for easier destruction. The other mechanism is agglutination. Agglutination is clumping together pathogens, okay, so that they don't move, and once they are clumped together, it is easier for them to be cleared. The fourth mechanism is complement activation, where there is a cascade of proteins that will be triggered to help to destroy the pathogens. This help happens through perforins, which help in perforating or creating pores on the membrane of the pathogens. Then another way is they do the granzymes. Granzymes are what lead to what we call apoptosis. Apoptosis is the programmed cell death. Okay, so a cell has its death programmed when the granzymes are created through the complement activation. The fifth is the antibody-dependent cellular cytotoxicity. So with this, infected cells are tagged for destruction by immune cells. So some cells which are infected are given a tag so that the specialized immune cells that will come and kill such cells, for example, the natural killer cell, will now come and destroy the cell that has been tagged, yenye imewekwa alama. Okay, so the antibody helps in such, in a mark a certain cell that needs to be destroyed. Then that cell will now be easier for the natural killer cells to come and identify which cell that needs to be killed. We've talked about immunologic memory now for some time, and the role of B cells with respect to the immunologic memory is once an infection is cleared, not all B cells resolve. Some of them change and they become memory B cells. After they've changed into memory B cells, they will persist in the body for some time. The importance of their persistence is to provide a response that is not only faster but stronger whenever there is re-exposure to the same antigen. And it is on this basis that now vaccination happens. Okay, so you have memory B cells. Then in case there is an exposure to the same antigen, your body, your immune system is now capable of providing a faster and stronger immune response, because a harmless version of the pathogen was introduced into the body. It is harmless because it cannot cause disease, but it is still immunologically viable, that it can trigger an immune response. Okay. So in summary, B cells play a key role in the immune system. They do so through the production of antibodies. The antibodies are the ones that will help to fight infections. For these B cells to act, they need to be activated, and that activation is done either through direct stimulation without the need of T cells. So we call that T-independent stimulation. Or it requires the assistance of T cells, the helper T cells or the CD4 cells, what we call now the T-dependent stimulation. These antibodies are Y-shaped. They have a variable region, they have a constant region, and they come with different classes. Okay, so you have some which deal with long-term immunity, there are others which deal with just allergic responses, and there are others which help with the mucosal protection. The antibodies work by eliminating pathogens, either through neutralization, or to neutralize is to block the pathogen from entering a cell. Opsonization, which is marking the pathogen for destruction. Then agglutination is binding the pathogens together. Once the pathogens are bound together, it will not only limit their spread, but it will make their likelihood to be cleared from the system easier. Then there is the complement activation, which happens by use of proteins that either make pores in the membrane of the pathogen, or they lead to apoptosis, which we call the programmed cell death. You have then the antibody-dependent cell cytotoxicity, which an antibody helps a specialized type of immune cells, the natural killer cells, to be able to recognize which cell needs to be killed. Okay, so the antibody will help in tagging a cell that is infected or abnormal. Being abnormal means it is potentially cancerous. So then the natural killer cells, or these specialized immune cells, can now come through and destroy that cell. The B cells will convert into plasma cells, and others will turn into memory B cells. The plasma cells help in stimulating the production of antibodies, but the memory B cells will remain in the body for long, so that they ensure there is a rapid and stronger immune response in case any previously encountered infection is re-exposed to your body. Okay, and therefore that is now what forms the foundation of vaccination. Okay. Yeah, so that's it about B cells and antibodies. Thinking about the structure of an antibody, it is Y-shaped. That is found on page 3. It has a constant region, it has a variable region. It is made up of a heavy chain and a light chain. At the top, at the tip of the variable region, that is where the antigen binds. That's where we have the antigen binding site. Okay. Yeah. So have a look at it. Otherwise, asanteni sana.